The next lesson on speciation. In this lesson, we're going to be looking at how evolution can occur through a species, and we're going to be looking specifically at isolating mechanisms known as allopatric speciation, which causes or results in the production of a new species. So natural selection is always the driving factor for evolution, so it will cause the evolution, or it will result in the evolution of a particular species, and it will do so by changing the allele frequency in the population. As we know, natural selection acts on the phenotypes of the organisms, which will promote evolution by selecting that specific favorable allele or favorable genotype, which will be passed on to the next generation. Before we begin, we're just giving a definition here of a species. A species is defined as a group of organisms or individuals who are genetically similar enough to reproduce with each one another, and they'll produce viable, fertile offspring. That means their offspring will live to an age. So viable means they can be born and they won't be a stillborn. Fertile means they can reproduce themselves. So viable, fertile offspring is what defines a species. If a species cannot interbreed, it means this can result in the production of a new species. Here is the formation or evolution of the human species. So isolating mechanisms. How do we have a new species created? Well, there are two types of isolating mechanisms. There's a prezygotic and postzygotic. So zygote refers to the egg. So prezygotic are mechanisms where organisms are isolated prior to the fertilization of the egg. Postzygotic are, of course, events where the zygote, which has already been fertilized, is now not able to develop into an organism. These are examples of prezygotic barriers, and these are examples of postzygotic, which we're going to go into. If all of these are avoided, we will have viable, fertile offspring produced. However, if these barriers take place, we'll not have the production of a viable, fertile offspring. So these are some examples of prezygotic and postzygotic barriers I'm going to go into now. So prezygotic barriers. We may have a geographical isolation of a species. They're separated by physical barriers. They cannot breed with one another. And over time, they'll have mutations, which means they can and are more and more dissimilar, and they cannot produce viable, fertile offspring. Maybe ecological isolation, where they're separated by their habitats. Or temporal. Birds migrate at different times of years, which means they no longer breed with one another. Or maybe even whales. Whales breed or migrate at different times of years, therefore they won't overlap, therefore they can't interbreed, so they'll be two separate species. Maybe behavioral isolation, where mating calls or the rituals they use to bring in the mate are so different that they cannot attract that mate, therefore they cannot breed to make viable, fertile offspring. Maybe structural or morphological differences. Differences in structure may mean that their reproductive organs can't be used to fertilize the zygote. It may be gamete mortality, so separated by fusing of gametes. So the gametes may not form a whole zygote. Some postzygotic mechanisms of isolation. We have where events prevent organisms from fertilizing each other to reproduce offspring. So for example, the horse and the donkey, they can reproduce and they will produce viable offspring, meaning the offspring, the mule, will survive, will get to the reproductive age. However, mules are unable to reproduce with each other. So a mule and another mule are unable to reproduce with each other, so they are not fertile. If they are not fertile, they are viable, they are still a different species. This demonstrates that these are different species because the offspring that's been produced cannot or is not viable and fertile. The reason they're not viable and fertile is because they have unequal pairs of homologous chromosomes, and if they separate by meiosis, it means there's going to be some cells which do not have the full number of chromosomes, which result in death of the embryo. So the process of speciation which we're required to know. The process of speciation is known as allopatric speciation. That's when a population becomes geographically isolated. So there's a geographical barrier or geographical isolation of that species, spatial barrier, it's also called, and separating subpopulations from interbreeding. So the process, we may have a large gene pool or a large population with a common gene pool. There'll be an isolating barrier. It may be a mountain, it may be a river, it may be the falling of a large tree or an object which prevents these two species from interbreeding with one another. Over time, they'll incur mutations. Those mutations will mean they are different, obviously, because they have different genetic profiles, genetic makeups. There'll be selection pressures which select for one particular phenotype, and that mutation may increase its allele frequency. Over time, these mutations mean these species or these populations can no longer interbreed to produce viable fertile offspring. That is defined as a new species. So the process, again, is shown over here. We have a single population. They are geographically isolated. They have different environments, and that will have different selection pressures, and those different selection pressures result in different phenotypes being favored, therefore different genotypes persisting, which means over time, those different genotypes will not be able to interbreed, and that will be defined as a new species. That is allopatric speciation. So the steps are: a population is divided, natural selection processes operate differently on these two regions, and there's no gene flow. Because if there was gene flow, that means the species will remain related. They'd share the same genes. Specifically, there has to be no gene flow between the two populations. Over time, random mutations will occur, and some of those mutations will be selected for, some of them won't. These mutations mean they're unable to reproduce to make viable, fertile offspring. And then that's the process for speciation. So let's look now at natural selection versus artificial selection. So natural selection is an example of that is allopatric speciation, or when favorable traits are chosen in a particular environment. Natural selection is completely random. It does not choose a particular trait because a human wants to have that trait. However, artificial selection is a human intervention for that trait. So we look here. The natural peach began 4,000 years ago, and that peach was around 25 millimeters. It had a 36% percentage of stone, and it was found in this area with 3 known varieties. Now, the artificial peach, so over time, humans have bred a particular peach where there's a reduction in the stone volume, a larger peach size, it's more soft and edible flesh compared to what was before. So the varieties have also increased. So over time, we have bred and selected for the favorable traits. Humans have done this. Another example, artificial corn. Corn has been chosen from at the beginning. Most of it was 19 millimeters. Now it's 10 times the size, easy to peel. Clearly, these traits have been chosen by humans, and this has not occurred naturally. So artificial selection is where humans intervene and choose the desirable traits. They're not for survival advantage, and at times they may be detrimental. The reason they may be detrimental is because these artificial traits may reduce genetic diversity. If we have a reduction in genetic diversity, that means that organism may be more susceptible to chance events such as disease. This is the summary of the lesson: natural selection and artificial selection, post-zygotic, pre-zygotic mechanisms of isolation, specifically allopatric speciation, and what that means and how it works. The key terms are written over here. These are the ones you need to be aware of this lesson. Thanks for watching. Be able to draw and define allopatric speciation with an example and describe pre- and post-zygotic isolating mechanisms for evolution, as well as describe the differences between artificial and natural selection. Thanks.